Okay, welcome back. In the last video, we finished the first part of our analysis of this kinematic data set. And now what we're going to do, we're going to take that data that we compiled from all 50 participants, and now we're going to work through that and extract some meaningful measures and do some basic statistical analysis and data visualization. So for this, we're going to go to the file that is called Analyze Joint Angle Data. And again, it's annotated and has a description of its purpose near the beginning here. All right, so I'll bypass this. So the first thing that we're going to do is we're going to load the file that we just created. Okay, so this was the summary data from our previous script. So I'm just going to highlight all this code, right-click, evaluate. So as we saw at the end of the last video, when we load that file, we end up with our average joint angle trajectories for the different tasks. So just to reorient you to the data that we're dealing with here, each of these variables has the same dimensions. This is a 3D array where each row corresponds to a different joint angle. So there are 12 joint angles. Each column corresponds to a different slice of the gait cycle where it's time normalized to have 101 data points. And then finally, each page, which is what I use to refer to that third dimension of the array, corresponds to a different participant. So we have 50 participants. So now what we want to do is to start extracting some data from this array. So the first thing that we're going to do is we're going to, as we mentioned at the beginning of the first video in this series, is that we're interested in understanding how some of the peak joint angles change across different tasks or different trial types. So the first thing that we want to do is extract these peak joint angles. So technically we're computing peaks of average joint angle trajectories. What you really want to do here is actually take the raw trajectories, compute the maxes from those, and then average those maxes. So just for simplicity here, this is what we're doing in this set of code. So here, lines 26 through 30 are where we do this. So let's walk through this code. So it's a little bit complex. So when you have a set of code that someone has given you, you want to figure out good ways to not just read the code to try to figure out what's going on, but to take the code and run small parts of it so that you can build an understanding. So as an example, I know that I have this variable in my workspace, average joint angles walking. We just talked about the dimensions of that variable. And so I can see in this line of code, if I scroll and highlight here, that here I'm extracting the 10th row for all of my participants. Okay. So now I can remember back to the last video and the last script that the 10th row of that data set corresponds to my ankle angle. Okay, so I just say, okay, that's ankle. So now what about this squeeze bit? So again, I'm working from the inside out as we've talked about in previous videos. So now I'm doing squeeze of that bit of code. So let's take this piece, copy, and we'll evaluate it directly. So I see something that is print to the workspace. It's not really, the command window, that's not really important. But here, I can see the dimensions, right? So I see it's 1 by 101 rows by 50 columns. Right? So it's the one row that I extracted from that data set. So what squeeze does is it basically collapses this to be a 2D array. Right? When I have if I have a 3D array where one dimension is 1, squeeze will just collapse this into a 2D matrix. So let's just see what happens here. So I highlight this code, everything up to this point, right click, evaluate selection. Again, it's printed to the command window, and now I can see I have a 101 by 50 matrix. And so now all of my rows correspond, or the rows correspond to different time points, points in the gate cycle, and the columns correspond to different participants. Okay. Lastly, I use max to find the max values, and max by default is going to work across for each column. So let's highlight this code, right click, evaluate selection. And again, I see now my answer is a 1/50 vector, where I have the maximum value for each participant. Okay. So again, this is something that you might want to pause the video for a bit and do the same thing that I was doing to make sure you understand, or not only what this code is doing, but you just get comfortable taking existing code and working through it by copying and pasting and running parts of the code. So that first line just computes my peak joint angles for normal walking, and then I do the same thing for all 5 of my other tasks. So when I run this block of code, what I end up with is a 5/50 matrix, where each row corresponds to a different task and each column a different participant. So that was for my ankle dorsiflexion or flexion. I basically do the exact same thing for my knee. The only difference is that I change my 10 to 7 so that I make sure that I'm extracting the correct part of my data set. Okay, so the next part is actually probably one of my favorite things to do, aside from coding in general and trying and manipulating data. Once we have the data in a form that we want to analyze, we often want to do that hand in hand with data visualization. So we've already used data visualization to just plot all of our raw data. That's really helpful. You should always do that just so that you actually build some intuition about the data that you're measuring. And next, what we're going to do is to take all of our data for each of our 3 different tasks that we're comparing. So walking, heel walking, and toe walking. And we want to look at the peak knee flexion, the peak ankle dorsiflexion values for all participants for those 3 tasks. So something that you might do if you're, no matter sort of how much experience you've had, it would be quite common for someone to take those data and for each task to report the mean and maybe the standard error, or if you're lucky, the standard deviation in the form of a bar and or an error bar plot, right? A bar plot with error bars. One of the problems with those types of plots is that they actually remove a lot of information, right? So here you're taking oftentimes a large data set and you're compressing it really to 2 numbers. Sometimes that's fine when your data have certain properties, but oftentimes in movement science specifically, we deal with relatively small data sets. And in those cases, it's often just best to present all the data. And so I'll show one way that you can do that. So here what we're going to use is a type of dot plot that in MATLAB at least is referred to as a swarm chart. So this is, I believe, a fairly new function. In the past, I and others have written custom functions to make these figures, but you'll get a gist of what it does briefly here. So again, what I like to do is to create my figure, give it a name or a label, a handle, and then once I do that, then I want to set some of the properties for my figure. So in this case, I'm going to save this figure as something that I would like to submit as part of a manuscript. So in that case, I want to specify the units of the figure. I change that to centimeters. And then position does 2 things. The first two numbers tell MATLAB where I believe it's the top left corner of the figure lives on your screen, so that positions your figure literally. Whereas the last two numbers are the height and width in centimeters. Sorry, the width and height, please. And so here I set these to be approximately what I would need if I wanted my figure to be one panel width in a standard journal article. And the reason why I do this is because if I make the figure the correct size now at the beginning, once I add the data and add my labels, if I can read it on the screen, it's probably good enough to be printed on a sheet of paper. All right, so what we're going to do here is use a function called swarm chart. Okay? So this is the dot plot that I was just referring to. So swarm chart has a few different arguments or inputs. And again, when we have a new function that we don't know, we can do a few things. We can do help, swarm chart. Okay. So typing in help in the command window is most useful for understanding the syntax of a function. So its inputs, the different options that you can set here. So for instance, the type of marker, colors, and so on. And then there's often simple examples. So let's actually look at what we can do when we're given an example like this. So I could actually just highlight this code, copy it, paste it, and actually I'm going to create a new figure first, paste it, press enter, and this is what I get. Okay, so this is a swarm chart. So the idea here is that you can plot lots of data for different conditions here. This would be 4 different conditions. And in places where the data have the similar y values, they're basically scattered along the x-axis. So this allows you to really get a sense of the distribution of your data. Right? So typically, if we have large data sets, we have most of the data near the middle of the distribution, and then fewer points near the extremes. All right, so let me close that. So let's go back here. So actually, let's go back here. So our swarm chart in this most basic operation has two inputs. The x values, and those are normally integers. So for instance, they correspond to different categories. And then the corresponding y values. So each data point has an x and a y coordinate. So in our case, I'm creating a vector that has our x coordinates. So let's first maybe look at our y coordinates. Okay. So I'm taking my peak ankle dorsiflexion variable that I created up here. I know the first 3 rows correspond to walking, heel walking, and toe walking. And so what I'm going to do now is extract each of those rows, and then I'm going to put them in an array here. Okay. So if I do this, copy, I just want to get the size of this. Okay. So because I'm putting them all side by side, I'm basically making a horizontally long or array or row vector. So again, walking would be my first set of points. Then my second would be, and I don't remember, heel walking and toe walking. All right. So now I need to create a vector of x values that correspond to those 3 tasks. And I want all the walking to correspond to 1, all the heel walking to correspond to 2, and then the toe walking to correspond to 3. I know I have 50 data points in each case because I have 50 participants. And so I'm going to use this function ones to create, in this case, a vector of 50 ones. So So if I just highlight this piece of code, again, this is what you should get into the habit of doing. I'm going to copy it, paste it in the command window, press enter. And so here what I can see now is that I have a set of ones, and I have, in this case, 50 ones. Then I go to 2, have some number 52s, and then change to 3. All right, so that's the most basic set of information that I need for my swarm chart. But we're going to specify a few different parameters or options. So in this case, instead of having open circles like the example we just saw, we want to use filled circles. We're going to specify something called the marker face alpha. So the alpha has to do with transparency. And so the face is basically the center part of the marker, and then the edge, of course, is the edge. All right, so that's gonna plot our data. Then here we're gonna, as we always do, specify where we want to place the tick marks, what we want those labels to be, and do the same thing for our Y label. All right, so let's highlight this block of code, evaluate selection, and there we go. So here is our figure. We see that on the x-axis we have each of our trial types. So walking, heel walking, and toe walking. Y values are our peak ankle dorsiflexion angles. We can see for walking, we tend to have peak angles that are on average maybe -10 degrees. For heel walking, right, you can imagine that when you're walking your heels, you dorsiflex, so you should have more positive angles. So here the mean is closer to 0. And then finally for toe walking, where now you would imagine you would be plantar flexed, you have much more negative values. Okay. So this is a way that we can show all the data. We can make sure that the results are consistent with our intuition or our understanding of mechanics, not really intuition here. And we can see how consistent these individual values are across participants for each task. So this is a really useful plot. The next thing that we'll do is that we'll actually run a repeated measures ANOVA. So just show you an example of one type of statistical test you could run in MATLAB. You could do most any other type of test that you would need. And then we'll, as you might do in a publication quality figure, we'll denote pairs of conditions that are statistically significantly different from one another. And then we'll export that as a PDF and save our results. All right, so I'll see you on our next and final video here. Actually, one last note is that we have another video that we've maybe referred to earlier that talks through some of the principles of data visualization. I highly recommend you check that out. It's linked in the GitHub, and that will really help you really think about different ways that you might visualize data and different factors and options that you want to set for your figures to really take control of the story you're communicating with your data. Okay, here we are in our final video, where we're going to do two main things here. We're going to perform a statistical analysis of our data. So we're going to run a repeated measures ANOVA to test whether there are tests for differences in our peak dorsiflexion angle across our 3 types of walking trials. Once we run that statistical analysis, we're then going to indicate the results directly on our figure using code, and then export that figure as a PDF. All right, so as I mentioned earlier in the previous video, you can perform most types of common statistical analyses directly in MATLAB through what's called the statistical analysis, or sorry, the statistics, I think it's the statistics and machine learning toolbox. So hopefully you already have this installed. If you don't, you may need to select add-ons and go to get add-ons to find this toolbox. Okay. So here in this section of code is where we're going to start our statistical analysis. So one of the first things that you need to do with most of these types of statistical analyses in MATLAB is reformat your data. Okay. So recently MATLAB has moved to a form where a lot of their statistical tools require you, or they operate on tables. So in this case, I'm going to create a table that contains my ankle dorsiflexion values, and I'm going to use the table function. So the table function has several arguments or inputs. The first is going to be or first set of inputs will be the values that you'd like to place in each of the columns. So each comma moves you to a different column. So in this case, my first column, I want to have the numbers 1 through 50. So this is going to correspond to my 50 participants. The second column will have each participant's peak dorsiflexion angle for normal walking. Then we'll do heel walking, so this is 2, and then finally toe walking. So after you've input the data, or you've specified all the variables that you'd like to input into your table, the next input or argument to the table function are options. All right, so in this case, the only option I'm specifying is the variable names. So this is basically, these will be the headers. This will be the header of my table. And I specify the names of the variables in each column as a cell array. Okay. So let's take a look at what happens here. So I'm going to highlight this bit of code, evaluate the selection, and then go take a look at this variable. Okay, so here's my table. Actually, let's do this. And so here again, I can see that I have my table, name of each of my variables. And one thing that I can see when I look closely at the table is that in some cases I have NaN values, and that would basically be a case where this participant 8, did not have any heel walking trials. All right. So, so now we have our table. So here we're going to do a repeated measures ANOVA. And so while this is not a course on statistics, even though if you'd like some more training on statistics or to develop your statistical analysis skills and expertise, we do have videos from Dr. Keith Lose that are based in R, but talk through a set of different types of statistical analyses. In this case, we're using a repeated measures analysis because for each person repeats different walking trials. And so essentially what we would like to do is to run an analysis that asks whether the dorsiflexion angle changes for each person on average as they move from one trial to another. So one thing we need to do to perform this analysis is to create a variable that contains information that specifies the trial type or the different conditions that the person performs. So here, this is just a vector that contains the numbers 1 through 3. Okay, so now what we want to do is fit our repeated measures model, and we're going to do so using a function called fit_rm. So I highly recommend that you go to the help page for this function, look at the repeated measures model class in MATLAB, and make sure that you understand how these models work and the various options that you need to set to sort of properly conduct these analyses. So here there are many different ways that we could do this analysis, but this is really more for the purpose of visualization. So when we run our analysis, our first input is the name of the table that we're going to use, the table that stores our data. The second input is the model that we're going to fit. Again, we don't have time to discuss this in great detail, unfortunately. But here, you can think about this tilde as an approximate equal. But the thing to keep in mind here is that on the left-hand side of the tilde, this is actually telling MATLAB that we're going to analyze all the variables from walking through toe walking. So in this case, we have walking, heel walking, and toe walking. And so we're going to model each of those and to look for differences between them eventually. Now, in this type of model, we could have both what we call between subjects factors and within subject factors. Here we're only interested in within subject factors, right? We don't have different groups in this study. We have a set of people who all do multiple tasks. And so we want to tell MATLAB with the within design option that condition is the variable we're going to use, so it has 3 levels. And we're going to look at all pairwise contrasts, right? So one walking and toe walking and heel walking, walking and heel walking. All right, so let's go ahead and fit our model. So I'll highlight this, evaluate. So let's take a look at this. So we'll clear our console, type repeated measures. So type the name of our model variable. And so now here we can see some information about the model and some of its properties. Okay. So next, when we actually want to do our first test, so we'd like to test to see whether there is any evidence to reject the hypothesis that all the conditions have the same mean. So we're going to run an ANOVA on our model. So what we'll do once we fit our model is that we'll, in this case, we'll just run an ANOVA and a multiple comparison. Again, this is really just to show an example of how we can take statistical results to illustrate them on a figure. But here we, by default, MATLAB refers to our variable as time. And so here, these are really our 3 conditions. And so each row corresponds to a comparison. So in this case, this would be comparing walking with, I believe, heel walking. And so the difference here would be column 1 minus column 2, so walking minus heel walking. The estimate of that difference, and then a test of the hypothesis that the null hypothesis that difference is 0. So as in all cases when we do statistics, we'd like to make sure that the statistics make sense, right? So do a sanity check. So for instance, if this says that 1 minus 2 on average should be about, or let's say condition 2 should have a dorsiflexion angle that's about 6.5 degrees greater than condition 1. I can hover over my figure. I see that from walking, my peak dorsiflexion is about -7. And so for heel walking, it should be near 0, and that's about where the midpoint is. Okay. Similarly, if I compare 2 and 3, right, so they should differ by about 25 degrees, where condition 2 is 25 degrees more than condition 3. So I already know this is approximately 0, and again, the mean here is roughly -25. Okay. So all of these comparisons, these differences pan out when I look at my actual data, right? So now I can be confident in my, at least this part of my statistics. So now what I would like to do is illustrate that each pair of conditions is actually statistically different from they're different from one another. And typically we'll do that by, drawing bars above each one and then adding an asterisk. We might do that in Illustrator or PowerPoint or some other application. So what we have here is I actually wrote some code that does this automatically. Okay. So I have a function. This is called a local function. We haven't really dealt with functions. That's maybe a little bit more advanced for some people. But a function is essentially a custom set of code that you can call and run many different times. So I've just created a function that I called significant difference. And it has, in this case, 6 inputs. First is that you have to tell MATLAB which axis you'd like to sort of annotate, which the X location, so the horizontal location of each group that you like to illustrate a comparison between. Y is the y-axis value where you'd like to place that bar. Height is a variable that dictates how tall you want those bars to be. And then symbol is the text you'd like to use to annotate, in this case, statistical significance. All right, so I won't walk through this in great detail. It's not really critical for this course, but you have it in case you'd like to use it. But all I do is use separate calls to the line function to basically draw on our figure. And then lastly, within this function, I use another call to text, which can add, here we're using it to add an asterisk, but you can also use it to annotate your figures and add information to make it easier for your reader or your audience to understand. Okay, so after we do our statistical analysis, we know that all pairs are different from one another. I then have a few different calls to my function significant difference, where I specify, the current figures axis, the pair of conditions I'd like to compare, where I'd like to place that comparison. So you can see once you start adding, in this case I start at a y value of 20, the next comparison I put a little bit higher, third comparison I put it a little bit higher than that, right? So it's a uniform distance between them so that things are symmetric and so on. Okay. So let's see what happens here. So I'm going to highlight this bit of code, right click, evaluate. Oh, I missed something here. Let's see. So unrecognized function. Yes. This is actually a good thing to illustrate. So one thing that can be challenging when you're running code that either you write or someone else has written is if I try to run this line of code, I received an error. When I tried to run it, I received an error. And that's because this function is a local function. So when I highlight the code and evaluate selection, MATLAB is looking for a function that lives outside of this particular script. All right, so I can't actually directly run it by just highlighting this code. That's okay. So what we'll do, we're actually just run the whole script. But before we do that, I just wanted to highlight these last two key lines of code where we save our figure as both a PDF here and a matlab.fig file. Okay. So hopefully you've already seen that, as we've done that in our previous videos for our model data analysis pipeline. So after that, we go through some of the same pieces of code for our knee flexion, but I didn't complete that. That's left for you all to complete, just to make sure you're getting a handle on how to do some basic processing and statistical analysis in MATLAB. So to conclude, I'm going to just press run and show you the final figure. So we'll minimize, this is the knee flexion one that's not complete. We minimize this. We now see our figure. It's the appropriate size, has our labels, indications of statistical significance. If I actually wanted to publish this, I'd make some maybe minor changes to some of the fonts and line thicknesses and things. But you can get the gist of it here. And if we go to our current folder, we can see our PDF. Right click, open outside MATLAB, and then here's our figure. Right, so this is the exact size that it would show up on essentially an 8.5 by 11 sheet of paper. Okay, so that concludes the walkthrough of our two example scripts here that we use to analyze the data sets for this part of the course. Again, they're going to be exercises that are described in the GitHub along with example code that you can complete yourself definitely take time to do that, and that's really one of the main ways that you're going to pick up the skills that we're introducing in this overall MATLAB introduction. As always, if you have questions, feel free to use our Repo Rehab Slack, contact your TAs or any of the instructors, and we can find a way to help you and get you up to speed.